Hello, this lecture is going to be the integumentary system, which as you can see is a fancy way to say skin. We're going to go over all the different parts, we're going to go over all the different types of terms, and again, if you just kind of think about this as what are the parts on this model, if I point to anything, can you tell, no matter which model I have, can you find these structures on these models? I'll kind of go through which models I'm most likely to use, but again, please use all the pictures that I have on Canvas and in this PowerPoint, just so you can be positive that no matter which view or which camera view, is it above or below or side or whatever, you know what these structures look like and you have a better understanding so you don't kind of have that stall or that brain fog during the midterm exam where you just can't come up with the answers. So let's get started. So there's only two layers of the skin, and you might have seen in that last picture epidermis and dermis. Even though the hypodermis it does have the name dermis in it, the hypodermis is below everything. So the epidermis is the top layer, and for that you're going to have to memorize the layers. So stratum basale, spinosum, granulosum, lucidum, and corneum are the terms you're going to have to memorize. For the dermis, you're going to have to memorize what are the structures. So what are the glands? What is the muscle? What are the different types of receptors called? The hypodermis, because it's below that, it's not part of the integumentary system. This is going to be a connective layer, which is going to have adipose and blood vessels. But we'll take a look at that when we do the test. One more thing before we kind of move on is you have two different types of skin. And you might sit there and be like, I have two different types of skin? Yeah, because if you feel your palm, you can feel your palm, it's kind of thick. Like even if you pinched it, take the back of your hand and pinch that, and that's going to be a different type of skin. The thick skin is going to have a special layer called stratum lucidum, and the stratum corneum is going to be a lot bigger, which makes it feel different. And you can pinch it, you see that it is much thicker than it is on the other part. And thin's going to not have the stratum lucidum and also have a smaller or narrower stratum corneum. You do have skin cells in the epidermis. So with those layers, you have stem cells, keratinocytes, melanocytes, and tactile and dendritic cells. The problem with these is if you just kind of look at all the models we have, all the models we have are pretty much gross anatomical models. So if you kind of looked at these, you can't really see the different cell types. All the models we have in lab, and I try to keep the online, virtual, or in-person all the same. I use the same models for all three of the modalities, but what I do is because we don't have a model with any of these, and you can see none of the models are going to have these cell types. So you can't really see stem cells undergoing mitosis. You can't really see brown cells in there that are going to be creating melanin, which is going to be that protein that's going to protect DNA, and it absorbs UV light. You also aren't going to have these types of cells that are going to be tactile cells. And what you will see on these is they have these little ridges that are in the dermis that are called the dermal papilla. And when we talk about the dermis, we'll talk about the dermal papilla, and we'll talk about touch receptors that are in there. But as far as tactile cells, you can't see them in the model, so that's why they're not in red. And you don't have dendritic cells, and these are just monocytes that have moved into the epidermis through blood and then have now just started to engulf and phagocytose bacteria and other types of debris within the epidermal layer. But what you're going to be doing is these things in red. So stratum basale is that first layer. And a good way to kind of think about it, basale, basement, bottom, that's how you know you're going to start with right above the dermal papilla is the stratum basale. Above that is going to be the stratum spinosum. And if you see, the further you get up in the stratum spinosum, you can see instead of being nice cubes or tall cells, they become very flat. And the reason why they're becoming flatter is because they're getting further and further away from the blood source. The blood's going to have the nutrients. The blood's going to have the oxygen. The further they get away from that, the more they die. And that's why by the time they get to the stratum granulosum, you start seeing these little dots of keratinohyaline pigment. And when they stain them, they stain dark purple. So they look like these little granules are in there, and that's going to be starting the process of water resistant for the skin. And because this is thick skin, you're going to have the stratum lucidum, which is that lighter layer. And then on top of the stratum lucidum is going to be the stratum corneum. And these next slides are just going to talk about what are some of the things. And the stratum basale, bottom layer, closest to the blood supply, so they're going to have a lot of different types of living cells. Stem cells are undergoing mitosis. They can undergo mitosis because what they do is then once they go through mitosis, they go up a layer into the spinosum, and then they keep migrating up. So the stratum basale is always producing new cells and pushing them up until they get to the stratum corneum. And if you just have like a black tabletop or even like a mouse or something like this, you can just take your arm, rub like this, and you'll see little pieces of your epithelium fall off or be exfoliated. And the next one is the spinosum, and remember, the further you get up, the flatter they become, because now they're starting to die and they're starting to shrink and they're starting to kind of get really, really, really skinny. The granulosum are going to have the 3 to 5 layers of keratinocytes. They're going to have that dark staining keratinohyaline granules, and that's how you can tell it apart, because it's usually going to be dark purple on no matter what model we go over. Then lucidum, like I've mentioned, only found in thick skin. It's usually going to be pale and featureless. That's why most of the models just have it as white if they have it. Then that corneum is usually going to be that upper layer above the granulosum in thin skin and lucidum in thick skin, and it's just going to be all dead skin. So if you just do this, kind of do this, and you feel it just like fluff off, it's going to be called exfoliation. That's why if you go into a spa, they use the pumice stone. They're just exfoliating your skin. They're removing dead layers of skin from the stratum corneum. And if you look right here, this is the model you see right here. So this model right here is very important because if you look at the bottom, you will see A, B, and C. A, B, and C corresponds to A is the hairless scalp. B, this middle layer right here, is the skin of the armpit. And then this one right here is going to be the sole of the foot. So what you're actually looking at right here is, if you see, you can see this layer right here is this part right here. So let's look at that. So I said B and C. You see a line right here. Everything over here is B. Everything over here is C. This is the foot, so you know that this is going to be thick skin. The armpit, if you wanted to touch your armpit, you could, and you would notice that it was thin. And this is going to be basale, stratum spinosum, stratum granulosum, stratum corneum. And then see the difference in thick skin: stratum basale, stratum spinosum, stratum granulosum with that purple, stratum lucidum is that white, and then that top layer is going to be the stratum corneum. So every time you see epidermis, no matter what model I have, here you can still clearly see the different layers of basale, spinosum, granulosum, lucidum, and corneum. So it's important to study the different models just so you get an idea. Start from the bottom, work your way up, and then that way you'll be able to tell, is there lucidum, is it thick or thin skin? Now we're going to the dermis. Every hair, or most hairs, are going to have a piloerector muscle, which is going to be associated with goosebumps. You're also going to have those finger-like projections that go up, which are going to be dermal papilla. So here are those finger-like projections that go up. So you can see the dermal papilla. And above the dermal papilla is going to be the epidermis. Then you have the hair follicle, which is going to consist of a bulb, a root, which is below the skin, and then the shaft, which sticks out. And then there's that piloerector muscle, or the erector pili muscle, which is going to give you goosebumps when it contracts. You have 3 main sweat glands you need to know: apocrine gland, sebaceous gland, and the meiocrine gland. Meacrine gland we're going to learn only in the axilla area, genital areas and stuff like that. Sebaceous glands are always going to be covering a hair follicle, and then the meacrine gland, apocrine gland is in the armpit, and bearded area of mature males and genital areas, and then the meacrine gland is everywhere. The meacrine gland, you know what your sweat tastes like. It tastes like salt, because your meacrine glands are always going to go to the surface of the skin, and then it's going to be that salty projections. So sebaceous gland, you're looking for a hair follicle. And one interesting thing to think about with the sebaceous gland. Sebaceous gland, if you work out or you're outside this weekend or whenever, you're outside and your hair gets kind of oily, that's going to be sebum, because sebum is going to be the secretion from the sebaceous gland. Sebum is not something that sounds like a good word, so marketers back in the 60s, they decided to say, well, we're not going to use sebum in our lotions. What we're going to do, we're going to brand it lanolin, because what happens with sheep also have sebaceous glands. Those sebaceous glands are going to create that oily secretion that goes onto their hair cells. The problem with sheep is they make wool. So the wool gets sheared off the sheep, the wool gets thrown into boiling, big vats of boiling water. It then gets pushed underneath the water, and then the fat, or the sebum, floats to the surface. They then collect that sebum off the surface of the water, they refine it, and call it lanolin. And then they put it in your moisturizers and creams, and usually the more expensive a moisturizer or cream is, the more lanolin it has, which is just sheep sebum. And if you're disgusted by that, and you go home and you look at your bottle and you're like, oh my God, I do have lanolin in it, that's going to be the secretion from the sebaceous gland onto a hair follicle of a sheep. The meacrine gland, like I said, is just going to be that surface of the skin. So if you're like, that tastes salty, it's just the sweat coming down and it's what you're tasting. If you look at your skin on a hot day, you can actually see the perspiration coming up, and that's why you can easily get the meacrine gland separated from the apocrine gland. The apocrine glands are going to be in the groin, anal, axilla, and armpit, and then areolar, bearded area of mature males. And I keep saying armpit because this is the model right here. That B is the armpit. So, that's your sebaceous gland. That is your apocrine gland right there, because it looks different. But I'll show you that on the model. So let's look at the difference. So you do have cremulus, which is going to be earwax, and the mammary glands, which we talk about in AMP2. But this is the first model I want to kind of bring up. Epidermis is going to be over here. Everything below that will be the dermis. And then you can see down here with the fat and blood vessels is the hypodermis. And one kind of way to think about it is, if you've ever seen a hypodermic needle, a very big needle, to puncture all the way down into that fat layer with the blood vessels. You do have those little finger-like projections, which are the dermal papilla. In the dermal papilla, I mentioned you do have touch receptors. You can call them tactile corpuscles or touch receptors. I don't care. Deeper down into the dermis, you do have something called pressure receptors, or lamellar corpuscles, or Pacinian corpuscles. I like pressure receptors because they're lamella, or they're laminated structures, so when they deform, they just touch the next layer, and then it sends an ion in, and then those ionic changes just send an action potential down the axon to tell the brain that there is pressure on certain parts of the skin. You do have the bulb, the root, and the shaft. You have the piloerector muscle that is attached to a hair follicle. You have the sebaceous gland that surrounds the hair follicle that's secreting the sebum. You have the meacrine gland that is going to the surface of the skin, apocrine gland, which is going to a nearby hair follicle. But here's the tricky part about that. I'll tell you about it on the next slide. But we covered everything. That was all the things we went over in this slide. On your midterm, there's anywhere from 2 to 6, sometimes even 8 questions on the skin, depending on how many models I'm going to use on your exam. And the reason why I said I'll get to the apocrine gland. Hair on top of your head, armpit, foot from earlier in the lecture. Because this is your armpit, That's your apocrine gland. You can see this green bottom based model. It's a company called Somso. They're German, and because they take kind of liberty, artistic liberty with some of these, that's why they labeled that the apocrine gland. So all of these white ones are going to be called meiocrine glands. Sebaceous glands are really clear because they're surrounding the hair follicle. You still have the bulb, the root, and the shaft. You have your piloerector muscle, or the erector pili, touch receptors up there, which are in the dermal papilla. And then down here, you can't see it because my face is in the way in the video. There's a pressure receptor or lamellar corpuscle, and you can see, if I can kind of do it, right there. So right where that dot is, that's going to be that lamellar corpuscle, Pacinian corpuscle, or touch receptor. And you might think, okay, what else? Well, if you go back and you go basale, spinosum, granulosum, lucidum, corneum, those are everything you need to know on this particular model. We do have other ones. So you saw me hold this one up earlier. This is the one up here in this corner. It does have the layers, where you can see the layers up here. It has the meiocrine glands. It has sebaceous glands. It has the pressure receptors down there. It's got touch receptors up here. So it's very easy. And it also has the dermal papilla in the middle. So you can use any of these models. Like if you look at all these models, sebaceous glands, you can see the pressure receptor, touch receptors, and dermal papilla will always be up here. If you look here, there's one right there, touch receptor. You do get the layers. This is going to have the stratum spinosum in there, which is that yellow layer right there. I don't know if you can see that, but if you look on top, you can see that it splits up the different layers for you. So that's one, and they have different sides of it, so you can see the receptors. And then finally, there's this model right here, which is very similar to the other ones. You can look at the piloerector muscle, sebaceous glands, meiocrine, pressure receptors, touch receptors, like that. And then I also have this picture, but I don't have it in my lab right now, where you can see the bulb, the root, the shaft. You can see the sebaceous gland, the piloerector muscle. You can see a bunch of meiocrine glands, pressure receptors, dermal papilla, stuff like that. So the only one you really have to worry about is every single model on this one, because it's such a good model and it's so unique because it has all the different structures. So please memorize and please go through all the different models. I have the videos, I have the pictures on Canvas. I do have most of the models in this PowerPoint lecture. So use the different models. Even though I'm more likely to use the German one with the green base on your test, it's important for you to look at it differently so you don't get stuck when you're taking the midterm. And good luck studying this material.